Hello everyone, myself Professor Shamsulayat. In today's discussion, we will explore the intricate role of ethylene, a gaseous hydrocarbon, in regulating plant growth and metabolism. Although ethylene is often linked to the ripening process, it also plays a role throughout the life of the plant. Let us begin with the historical background of ethylene. In 1864, it was observed that gas escaping from street lights resulted in inhibited growth, distorted plant formation, and unusual thickening of stems. The first insight into the discovery of ethylene can be traced back to George Feinstock 1858 report, which highlighted that the release of illuminating gas, commonly employed for lighting in household and street lamps during that period, inflicted harm on plants. In 1896, Neil Jobo from the University of St. Petersburg, Russia, identified ethylene as the active component in the illuminating gas which affect plant growth. Later in the year 1901, they observed a triple response of pea seedlings which was induced by ethylene. Cousins in the year 1910 was the first to indicate that plant biosynthesized ethylene, as he reported that fungus damaged oranges produce a gas which accelerated fruit ripening in banana. Richard Gane in the year 1934 estimated ethylene released from apples and demonstrated that plants biosynthesize ethylene. Finally, in 1935, Crocker and his co-worker proposed that ethylene is a plant hormone. Our current understanding of ethylene receptors has been obtained from the characterization of ethylene insensitive mutants. Later, in the year 1970, Young and co-workers elucidated ethylene biosynthetic pathway. Moving ahead to ethylene chemistry. Ethylene is an unsaturated hydrocarbon, or we can also call it simplest alkane having formula C2H4. Moreover, it is a colorless gas which is lighter than air at room temperature and sparingly soluble in water. Ethylene is biologically active at very low concentration, which may vary from 0.1 to 1 ppm. Moreover, lower or higher sensitivity is also observed depending upon the plant species and their response. In 1901, Neil Jobo observed that ethylene offers, or you can say the ethylene alters the growth of etiolated pea seedlings and leads to shorter, thicker hypocotyl which display a diageotropism. This was the first biological effect of ethylene reported in plants and became subsequently known as the triple response of etiolated seedlings in response to ethylene. Ethylene is produced in fungi, bacteria and higher plants, with all living cells possessing the ability to produce this compound. Higher level of ethylenes are synthesized in meristematic tissues, peripheral tissues and nodal regions. Dormant apple buds and ripening fruits exhibit elevated ethylene synthesis. The production of ethylene is increased by actions such as plucking, cutting or injuring tissues. The transport, how they are going to be transported? As you know that ethylene is introduced into the atmosphere from physical sources such as combustion gases from the kerosene store, tobacco smoke, car exhaust as well as plant sources. Ethylene is extremely volatile and can freely diffuse through lipid membranes. Additionally, ethylene produced in a particular location can be detected by cell from both distant from its point of synthesis and within the same cell without the requirement of a transporter. Here in this figure, the synthesis of ethylene in plant is described. The synthesis of ethylene in plants basically involves a three-step processes that originate from the methionine. So, methionine is a precursor. Firstly, methionine is transformed into S-adenosyl L-methionine called SAM through the activity of SAM synthetase. Next, SAM undergo conversion into 1-aminocyclopropane 1-carboxylic acid called ACC, catalyzed by ACC synthase, also known as ACS. In the final step, ACC is transformed into ethylene with the assistance of ACC oxidase called ACO. ACS is triggered by various factors such as stress and fruit ripening. Additionally, the turnover of ACC synthase, crucial for regulating ethylene biosynthesis, is controlled through phosphorylation. ACC synthase is an unstable enzyme and is encoded by a multi-gene family, which is differentially regulated by various inducers of ethylene biosynthesis. In plants, methionine is present in limited amount and hence is regenerated through a cycle called Young cycle. Without this cycle, the amount of reduced sulfur present would limit the available methionine and also the synthesis of ethylene. Thus, during ethylene production, methionine depletion is prevented due to recycling of methionine. ACS catalyzes the rate limiting step in ethylene biosynthesis, and ACS enzyme levels are controlled by transpiration and through protein stability. ACC oxidase catalyzes the last step in ethylene biosynthesis. In tissues that show high rate of ethylene production, ACO activity can be a rate limiting step during ethylene biosynthesis. This enzyme requires iron and ascorbate for its activity. This slide shows the mechanism of ethylene. Basically, the mechanism of ethylene involves its dissolution in cell membrane, leading to a modification in their permeability. Substantial evidence suggests that the hormone binds to a metal, potentially copper, causing an alteration in an undefined manner. Silver functions as an antagonist to ethylene action, likely competing with copper for the receptor. Ethylene is recognized as a suppressor of genes involved in the production of mRNA responsible for synthesizing various enzymes such as cellulase, protease, alpha amylase, catalase, peroxidase, and invertase. The application of actinomycin D and obligor cycloheximide disrupt the de novo synthesis of these enzymes, consequently delaying the abscission and senescence of organs. This slide gives a brief introduction of the physiological role of ethylene. As you can look, ethylene regulates several physiological processes in plant such as ripening of the fruit, breaking seed dormancy, apical dominance, growth inhibition, accelerate the abscission, inhibit flowering, epinasty, and sex expression, as well as pulmonar hook formation. Now, we will discuss one by one each physiological responses regulated by ethylene. This slide shows the response of ethylene in relation to fruit ripening. Fruit ripening is a complex process that involves softening, color change, and change in chemical content. The disappearance of organic acids, solubilization of middle lamella, and cell wall polysaccharides, as well as hydrolysis of starch and fats. Therefore, ethylene is known as fruit ripening hormone since it is involved in the ripening of fruits. Different types of fruits react differently with the exogenous application of ethylene. Oxygen is necessary for ethylene action, and carbon dioxide inhibits the action of ethylene. Commercially, gas storage of fruits involves placing them in a sealed room at a low temperature. The oxygen level naturally decreases due to respiration, leading to an increase in carbon dioxide. To mitigate the ethylene produced by fruit, it is absorbed using brominated charcoal filters. Ethylene also play a role in breaking the dormancy of many seeds such as cereals. It also used to promote bud sprouting in potato and other tubers. Moving to next, the physiological role, that is apical dominance. It is maintained through the inhibition of lateral bud growth by ethylene, similar to the role of auxin. Ethylene serves as the secondary messenger for the primary stimulus of IA in the regulation of apical dominance. This slide indicates the role of ethylene on growth and epinasty. The ethylene hinders the lengthwise expansion of the stem. However, it induces lateral growth, increasing stem girth. The lateral swelling is a result of cell enlargement rather than cell division. The inhibitory effect of ethylene on plant growth is temporary and normal growth resumes when ethylene is eliminated. Additionally, the application of carbon dioxide can overcome the inhibitory impact of ethylene. Ethylene also restrains the elongation of internodes. Now we will discuss another important physiological response, which is epinasty. The epinasty is also called as leaf bending. Epinasty is characterized by the swelling of cells on the upper section of the petiole, leading to the drooping of leaves. Monocots do not exhibit epinasty. Young leaves are more susceptible to this process compared to older ones. The occurrence of epinasty may be attributed to the migration of auxin towards the upper side of the petioles. An increased auxin concentration causes the elongation of cells on the upper side, resulting in the epinasty. This slide shows the response of ethylene to flowering, sex expression as well as the abscission. Ethylene has a widespread inhibitory effect on the flowering in plants, with the notable exception of pineapple, plumbago and cucurbits. Interestingly, ethylene can stimulate flowering in short day plants even under long day condition. Application of ethylene diminishes the formation of male flowers while promoting the development of female flower. Consequently, the use of ethylene results in increased fruit production, albeit with smaller fruit size, as seen in crops like cucumber and pumpkin. Moreover, ethylene increases the shedding of both vegetative parts such as leaves and stems and reproductive organs like flowers and fruits in plants. Its primary role involves inducing senescence in leaves and initiating the activity of cell wall degrading enzymes in the abscission region. Additionally, ethylene hinders the access of oxygen to the abscission zone, leading to reduced oxygen level in that region. Ethylene maintains plumular hook, plumular tip, that is shoot apex, is usually bent like a hook, which is advantageous to seedling for penetration through the soil, protecting the tender apical growing point from being injured. The last one is the ethylene signaling. Here we will discuss the ethylene signaling in Arabidopsis as displayed in the figure. The ethylene receptors such as ETR1, which is situated on the endoplasmic reticulum membrane and participate with two downstream signaling proteins. They are CTR1, also called constitutive triple response 1, and EIN2, that is ethylene insensitive 2. CTR1 functions as a soluble serine threonine kinase and maintains a physical association with ethylene receptors. EIN2, on the other hand, is an ER transmembrane protein featuring a cytosolic C-terminal domain that serve as a target of the CTR1 kinase activity. So, EIN plays a crucial role in stabilizing transcription factors belonging to the EIN3 called ethylene insensitive E3 family, which in turn activate the transcription of genes responsive to ethylene. In the absence of ethylene, the ethylene receptors act as a negative regulators, actively suppressing the hormone response. When ethylene is absent, the ethylene receptors stimulate the CTR1 kinase, leading to direct phosphorylation and subsequent inactivation of the EIN2. Active CTR1 functions as an additional negative regulator in the ethylene response pathway. The binding of ethylene to the N-terminal transmembrane domain of the ethylene receptors results in their inactivation and the deactivation of CTR1. This process involves the dephosphorylation of EIN2 by an unidentified phosphatase, followed by the proteolytic cleavage of its cytosolic C terminus by an unidentified protease. The interaction between CTR1 and EIN2 along with similar proteins regulate the receptor's ability to ensure the swift reset of ethylene response mechanism. Subsequently, the released EIN2 C-terminal domain translocate into the nucleus, where it activates EIN3 either directly or indirectly. The activated EIN3 family of transcription factor coordinates the transcription of numerous genes rapidly induced by ethylene, including the key transcription factor ERF1, that is ethylene response factor 1. The activation of EIN3 and the ERF bring about a significant change in the expression of multiple genes, ultimately influencing the functionality of the plant cells in response to ethylene. So, this is all about the signaling of the ethylene. As we conclude today's session, we have covered ethylene biosynthesis, its physiological response, and the intricacies of its signaling pathway, from its role in ripening to influencing flowering, sex expression, and abscission